Hello everyone, my name is Shubham Varadkar. I am a final year B Pharmacy student in Shivajirao as Jondhle College of Pharmacy, Asangao, and I am going to explain about floating Gastro Retentive system along with my coordinator Yadnesh Marade in the guidance of Mrs. Gita Mohire, who is Professor of Pharmaceutics in our college, and Dr. Nitin Mohire, Principal of our college. So let us see what is Gastro Retentive system. Gastroretentive drug delivery is an approach to prolong gastric residence time, thereby targeting site-specific drug release in the upper gastrointestinal tract for local and systemic effects. So, this is the system in which the drug stay in our body for long time and hence its duration of action also gets increased. And this system is usually applied for the gastrointestinal tract disorders. Gastroretentive forms can remain in the gastric region for long periods and hence significantly prolong the gastric retention time of drugs, because of which the drug remain in our body for longer time and show effects. This system also enhances the bioavailability and controlled delivery of many drugs. Usually, there are many drugs which release and metabolized in our body very quickly. And by applying gastroretentive system, those drugs release in our body slowly for a particular time interval. The concept involved in GRDDS is increasing the gastric retention time. Gastric retention time is the time in which the drug stay in the gastric region of our body. GRDDS help to increase this time. Drugs which require increase in bioavailability and controlled delivery can also be formulated by utilizing the novel concept GRDDS. Now let us see the need for gastroretention. Gastroretention is needed for the drugs that are absorbed from the proximal part of the gastrointestinal tract. It is also needed for drugs that are less soluble or that degrade at alkaline pH, drugs that are absorbed due to variable gastric emptying time, and treatment of peptic ulcer caused by Helicobacter pylori infection. Stomach. The stomach is situated in the left upper part of the abdominal cavity immediately under the diaphragm. Its size varies according to the amount of distension. Up to 1500 ml following a meal after food has emptied, a collapsed state is obtained with resting volume of 25 to 50 ml. The stomach is anatomically divided into three parts, that is fundus, body and antrum. The proximal stomach, made up of fundus and body regions. Proximal means the point of attachment where the stomach is attached to the esophagus. It serves as a reservoir for the ingested material, while the distal region, that is antrum, is the major site of mixing motions, which act as a pump to accomplish gastric emptying. Now let us see the diagram of stomach. The stomach is divided into three parts, that is fundus, body and antrum. The fundus is connected to esophagus through gastroesophageal junction. What are the function of these parts? We will see that. Fundus. Fundus stores undigested food and gases that are released during the process of chemical digestion. Body. Body is the largest region of the stomach that is found between fundus and antrum. Antrum holds the broken food until it is ready to be released into the small intestine. Pylorus. Pylorus is the last part of the stomach which acts as a valve to control the emptying of the stomach contents into the small intestine. Its distal end, that is pyloric sphincter, is connected to duodenum of small intestine. Lesser curvature. It is short, therefore it is known as lesser curvature. It is concave curve on the right from the opening of esophagus to the opening into the duodenum and lies on the posterior surface of stomach. Greater curvature. It is 4 to 5 times as long as lesser curvature and convex curve on the left from the opening of esophagus to the opening into the duodenum. Drug candidates for GRDDS. Narrow absorption window in GI tract. Example, riboflavin and levodopa. Primarily absorbed from stomach and upper part of GIT. Example, calcium supplements, chlorodiazepoxide and cinnarizine. Drugs that act locally in the stomach. Example, antacids and misoprostol. Drugs that degrade in the colon. Example, ranitidine, HCl and metronidazole. Drugs that disturb normal colonic bacteria. Example, amoxicillin trihydrate. Floating drug delivery system. Floating drug delivery system was first described by Davies in 1968. It have a bulk density less than gastric fluids. Low density system that have sufficient buoyancy and so remain buoyant in the stomach without affecting the gastric emptying rate for a prolonged period of time. While the system floats over the gastric contents, the drug is released slowly at the desired rate, which released in increased GRT and reduces fluctuation in plasma drug concentration. Advantages of FDDS. It improves patient compliance by decreasing dosing frequency. Better therapeutic effect of short half-life drugs can be achieved. Gastric retention time is increased because of buoyancy. Drug releases in a controlled manner for a prolonged period. Site-specific drug delivery to stomach can be achieved. Enhanced absorption of drugs which solubilize only in the stomach. Superior to single unit floating dosage forms as such microspheres releasing drug uniformly and there is no risk of dose dumping. Limitations of floating drug delivery system. High level of fluids in the stomach is required for maintaining buoyancy and thus working of FDDS. Not feasible for drugs having solubility or stability problems in gastric fluid. Drugs such as nifedipine, which is well absorbed along the entire GIT and which undergoes significant first pass metabolism, may not be desirable candidates for FDDS since the slow gastric emptying may lead to reduced systemic bioavailability. Limitations to the applicability of FDDS for drugs that are irritating gastric mucosa. Classification of FDDS. Floating drug delivery system is classified into two types. They are non-effervescent systems and effervescent systems. Non-effervescent systems consist of subtypes of colloidal gel barrier system, microporous compartment system, alginate beads and hollow microspheres. Effervescent system consist of volatile liquid containing system and gas generating system. This type of system, after swallowing, swells via imbibition of gastric fluid to an extent that it prevents their exit from the stomach. Non-effervescent systems incorporate a high level, that is 20 to 75% weight by weight of one or more gel forming highly swellable cellulosic hydrocolloids. Example, hydroxyethyl cellulose, hydroxypropyl cellulose, hydroxypropyl methyl cellulose and sodium carboxymethyl cellulose. Polysaccharides or matrix forming polymers. Example, polycarbophil, polyacrylates and polystyrene into tablets or capsules. On contact with gastric fluid, these gel form or polysaccharides and polymers hydrate and form a colloidal gel barrier that controls the rate of fluid penetration into the device and consequent drug release. As the exterior surface of the dosage form dissolves, the gel layer is maintained by the hydration of the adjacent hydrocolloid layer. The air trapped by the swollen polymer lowers the density and confers buoyancy to the dosage forms. As a result of this, the dosage form floats in the gastric fluids and release the drug. Colloidal barrier system. This system contains drug with gel forming hydrocolloids meant to remain buoyant on the stomach content. This prolongs gastro retentive time and maximizes the amount of drug that reaches its absorption sites in the solution form for ready absorption. This system incorporates a high level of one or more gel forming highly soluble cellulose type hydrocolloid. Example: hydroxypropyl cellulose, hydroxyethyl cellulose, hydroxypropyl methyl cellulose, polysaccharides, and matrix forming polymer such as polycarbophil, polyacrylate, and polystyrene. On coming in contact with gastric fluid, the hydrocolloid in the system hydrates and forms a colloidal gel barrier around its surface. This gel barrier controls the rate of fluid penetration into the device and consequent release of drug. So friends, generally in this system, the drug is covered with gel forming hydrocolloidal layer, because of which the dosage form remains buoyant. Buoyant means it floats on the gastric fluid. And after reaching the stomach, when the dosage form comes in contact with gastric fluid, the hydrocolloid in the system hydrates and forms colloidal gel barrier. Because of this colloidal gel barrier, there is controlled fluid penetration and therefore controlled drug release. HBS system, also known as hydrodynamically balanced system. So HBS are best suited for drugs having a better solubility in an acidic environment, and also for the drugs having a specific site of absorption in the upper part of the small intestine. HBS system can remain in the stomach for long periods and hence can release the drug over a prolonged period of time. Hence, the problem of short gastric residence time encountered with an oral controlled release formulations can be overcome with this system. These systems have a bulk density less than gastric fluid, as a result of which they can float on the gastric contents. These systems are relatively large in size and passing from the pyloric opening is prohibited. So, as we see in the diagram, when the dosage form comes in contact with gastric fluid, the hydrocolloid layer hydrates and therefore there is drug diffusion in the stomach. This drug diffusion is because of the erosion of gel barrier. The HBS must comply with three major criteria. It must have sufficient structure to form a cohesive gel barrier. It must maintain an overall specific density lower than that of gastric contents. It should dissolve slowly enough to serve as a reservoir for delivery system. Alginate beads. These are multi-unit floating dosage forms have been developed from freeze-dried calcium alginate. Spherical beads of approximately 2.5 mm in diameter can be prepared by dropping sodium alginate solution into aqueous solution of calcium chloride, causing the precipitation of calcium alginate. The beads are then separated, snap frozen in liquid nitrogen, and freeze dried at -40 degrees Celsius for 24 hours, leading to the formation of a porous system which can maintain a floating force for over 12 hours. These floating beads gave a prolonged residence time of more than 5.5 hours when compared with solid beads which gave short residence time of 1 hour. Hollow microspheres or microballoons. Hollow microspheres loaded with drug in their outer polymer shelf were prepared by a novel emulsion solvent diffusion method. The ethanol dichloromethane solution of the drug and an enteric acrylic polymer was poured into an agitated solution of polyvinyl alcohol that was thermally controlled at 40 degrees Celsius. The gas phase is generated in the dispersed polymer droplet by the evaporation of dichloromethane formed an internal cavity in the microsphere of the polymer with drug. The microballoon floated continuously over the surface of an acidic dissolution media containing surfactant for more than 12 hours. Now let us see the process in which firstly there is the formation of emulsion, then there is formation of shell around the emulsion, then there is generation of gas phase, and at last the formation of microballoon. Now let us understand the mechanism behind the formation of microballoon. Within oil and water emulsion, there is rapid diffusion of ethyl alcohol, because of which there is shell formation in which the drug is loaded. Further, there is evaporation and diffusion of dichloromethane, because of which there is hollow space created within the center of the shell, and there is the formation of hollow sphere which is responsible for the buoyancy of the dosage form. Microporous compartment system. This technology is based on the encapsulation of a drug reservoir inside a microporous compartment with pores along its top and bottom walls. The peripheral walls of the drug reservoir compartment are completely sealed to prevent any direct contact of gastric surface with the undissolved drug. In the stomach, the flotation chamber containing entrapped air causes the delivery system to float over the gastric content. Gastric fluids enter through the aperture, dissolves the drug, and carries the dissolved drug for continuous transport across the intestine for absorption. The diagram contains microporous wall at upper surface and lower bottom, a flotation chamber in which the air is trapped, because of which it floats on the gastric fluids, a drug reservoir, and the peripheral sides are sealed. The gastric fluids enters through the aperture of this microporous valve, dissolve the drug, and carries the dissolved drug for continuous transport across the intestine for absorption. Effervescent systems. We will see about the subtype of effervescent system, that is volatile liquid containing system. The GRT of a drug delivery system sustained by incorporating an inflatable chamber which contains a liquid, example ether, cyclopentane, that gasifies at body temperature to cause the inflation of the chamber in the stomach. These devices are osmotically controlled floating system containing hollow deformable unit that can convert from a collapsed to an expanded position and returns to collapsed position after an extended period. The deformable system consists of two chambers separated by an impermeable, pressure responsive, movable bladder. The first chamber contains the drug and the second chamber contains volatile liquid. The device inflates and the drug is continuously released from the reservoir in the gastric fluid. The device may also contain a bioerodible plug made up of PVA, PE that gradually dissolves, causing the inflatable chamber to release gas and collapse after a predetermined time to permit the spontaneous ejection of the inflatable system from the stomach. Gas generating systems. These buoyant delivery systems utilize effervescent reaction between carbonate, bicarbonate salts and citric tartaric acid to liberate CO2, which gets entrapped in the jellified hydrocolloid layer of the system, thus decreasing its specific gravity and making it float over chyme. These tablets may be either single layered wherein the CO2 generating components are compressed in one hydrocolloid containing layer and the drug in the other layer formulated for a SR effect. When the system comes in contact with gastric fluid, the sodium bicarbonate and tartaric acid within the system react to liberate carbon dioxide, which pushes the polymer layer outside and allows the system to float over a chyme by reducing its specific gravity, and thus drug diffusion takes place. These are some marketed preparations of FDDS. We are thankful to the President of our college, Dr. Shivajirao S. Jondhle sir, Secretary, Dr. Geeta Khare ma'am, and the Principal of our college, Dr. Nitin Mohire sir. Hope you like this video. For more informative videos, subscribe to our YouTube channel.